So today we're going to move on and talk about some of the genetics part of Science 30 course, looking at how your traits are inherited, how disease can be inherited, passed along. There are some differences between Bio 30 and Science 30 here. We only look at, in Science 30, some really basic, called monohybrid crosses, where we look at one trait at a time, and sex-linked crosses, traits that are passed along on the X chromosome. All of the other ones that you would have done in Bio 30 if you took that course aren't covered in this course. So some differences between the way you inherit traits or pass traits onto your children. There's a difference between acquired traits and inherited traits. Acquired traits, as the name kind of suggests, are things that are acquired during your lifetime. So things like your different experiences like travel, your education, how you were raised. You know, this would be things like maybe, for example, racism, scars, ability to speak a language. Those are all things acquired during your lifetime. Those are things that aren't passed on, obviously, to your children. Obvious to us today, if you took Science 20 or Bio 20, you would have learned about a guy named Lamarck who thought that acquired traits were things that were passed on to our offspring, and that's how evolution happened. But that was proven to be incorrect. Inherited traits are specifically found, or traits found on our genes, on our chromosomes. So things like blood type, eye color, diseases, our shape of our nose, all of those types of things are inherited traits. A term that we use to describe the genes itself, or alternative expressions of the genes, are called alleles. And if we look at, let's just say, for example, this is chromosome number 7. Let's say there's a certain gene on chromosome number 7, maybe for a disease or something. There could be a gene on one of the chromosomes that you got from one of your parents that would be for, say, normal trait, and then the gene on the other chromosome that you got from your other parent would be maybe for a recessive trait that causes the disorder. So rather than us talk about maybe, for example, normal skin pigmentation and albinism, we would say, you know, big A, maybe if we were using albinism as the example, and little a. Those would be the alleles that represent those traits. Now, when we look at the actual genetics part, we'll talk about dominance and recessiveness. Now, dominant traits are caused by dominant alleles, okay, those letters, and that's expressed by a capital letter. So a capital A, for example, would represent the dominant allele for that trait. If normal, if we stick with the normal skin pigmentation, and for albinism, or for not having albinism, a big A would be for normal skin pigmentation, whereas a little a, we'll get to next on recessive, would be for the albinism trait. Now, what's key here is as long as you have one allele, at least one dominant allele, you're going to have the dominant trait. So for example, try and draw this out. If, because your alleles are always in pairs, if you had a dominant allele from one parent and your other parent gave you a dominant allele, you would have the dominant trait. But if you have one dominant allele and a recessive allele, you still get the dominant trait. So the rule is, as long as you have one capital letter, one dominant allele, that dominant trait is what the person would possess. Now, the only way you're going to get the recessive allele then, or the recessive trait, I should say, is if you have two of the recessive alleles. So if we go with the albinism trait again, the only way we are going to get albinism is if you have a little a, you have two recessive alleles, because the rule again was if you have one dominant allele, one capital letter, you get the dominant trait. Now, a cool way for us to be able to show what we call probability, because that's what we're always interested in genetics. What is the probability of children from parents that have these particular gene combinations? What's the probability of their children having a certain trait? And we use what's called a Punnett Square to be able to do that. Now, to create a Punnett Square, it's actually quite easy. We just make a little square. That should look like a square. We put a little cross through the middle of it like that. Sorry about that, a little mess right there. And what we can do is we can take whatever the alleles are from the parents and we can put them in these spaces right here. So for example, let's say we took one parent that had that and another parent that had this, okay? Big A, little a, little a, little a. Again, if we go with the albinism, This would be, hopefully you remember, not have albinism because they have the dominant gene for not having it, and this would be a parent that had it. So what we will do is we'll take the big A, put it there, the little a, put it there, and then we take the other parent, the little a, and the little a, and we drop it into those two spaces. These spaces represent what the gametes would be for each of those parents. And then what we do in the boxes here is we show the combinations. So for example, if this was the male, this is maybe the sperm, and these are the eggs. If that sperm fertilized that egg, in this box, we're going to get a big A, little a. In this box, we'll get a little a. If you can see that, little a, little a. This box, if that sperm fertilizes that egg, we go big A, whoops, little a. And in the bottom box, little a and little a. Tough drawing with the mouse. Now, the probability here is what we're looking for. So a question may say, what is the probability that a parent who has the albinism gene but doesn't have albinism and has children with a parent who has albinism, what is the probability that their children will have albinism? And if we look at all these boxes, you have to be able to read this and see that 1, 2 out of 1, 2, 3, 4, 2 out of 4 would have albinism, and you'd be able to determine that we've got a 50% probability of the children having albinism. Now remember, this is just a probability situation. So just because you had one child that had albinism, that doesn't guarantee that the next child isn't going to have it. Every single time a child is produced by that couple, there's a 50% probability. Okay. Now there's some other terminology here that you need to know. I mentioned in the last one like big A, little a, and little a, little a. On an exam, we're not going to tell you that the parents are big A, and big A, little a. What we'll do is we'll use words like homozygous or heterozygous. For homozygous, there's two different examples. There's big P, for example, and little p. So two dominants or two recessive. We would call this one homozygous dominant, homozygous meaning the same. This one would be homozygous recessive. And then for here, if the individual was heterozygous, big P, big A, little a, whatever, heterozygous meaning different, they've got one of each allele. Remembering that this individual and this individual would both have exactly the same traits though, physical expression or the trait of that gene, because they both have at least one of the dominant alleles. Okay, now moving along here a little bit, there's some other terminology, excuse me, phenotypes and genotypes. So when I describe the actual trait, I'm talking about the phenotype of that individual. So if I said somebody had albinism, or they had normal skin pigmentation, or they had hemophilia, or their blood type was blood type A, that would be describing their phenotype. Think pheno, physical, right? It kind of starts the same. So this is a physical description of the trait. Whereas genotype looks at the actual genes. It looks at the letters or the alleles. So if I said an individual was big A, that would be its genotype. Or big A, little a, or say heterozygous, that would be the individual's genotype. So these are things you got to watch for on an exam. You need to watch for the homozygous and the heterozygous term. You'll need to watch for whether the individual is dominant, or sorry, the trait is dominant or recessive. And then you'll have to watch for whether the question is asking about the phenotype or the genotype of the offspring. Now there's another type of genetic test or genetic cross that we can do, which doesn't involve just looking at the big A's and the little a's. That would be if a gene was found on chromosome number 1 through chromosome number 22. Remember, there was a special pair of chromosomes, chromosome pair number 23, and those were the sex chromosomes. So in this diagram here, we've got an X chromosome, and here we've got a Y chromosome. Remember the difference between the X chromosome was bigger, the Y chromosome is smaller. Now if you look at this one has a lot more genetic material on it than this one does. The genes found on the X chromosome are not the same as the genes found on the Y chromosome. So that's going to be key. Remember that X chromosome carries certain genes, the Y chromosome carries certain genes. So when we talk about this next type of genetic cross called sex-linked traits or sex-linked inheritance, it's important to note that in Science 30, we're only going to be dealing with ones found on the X chromosome. There are genes found on the Y, but that's only going to affect half of you guys because only half of you guys have a Y chromosome, right? Only the boys, only the males in the class would have the Y chromosome. Now, if we look at gender determination, here would be your mom, here would be your dad. Just to show how we get that roughly equal 50-50 split between having children. There are the X's that go into mom's cells, her eggs. Here are the X and the Y's. If that sperm fertilizes that egg, we get a female. That sperm fertilizes that egg, we get a female, and so on. If that sperm fertilizes that egg, we get the males. So a 50-50 chance between producing the two. Now with these sex-linked disorders, unlike the autosomal disorders that we talked about, which was on chromosome number 1 through chromosome number 22, we also have these sex-linked inheritance. So these would be things that are not primary or secondary sex characteristics. Okay, nothing to do with organs that make you look male or female, having a deep voice, having hair, etc. Not related to that at all. Sex-linked inheritance is things like hemophilia, colorblindness, etc. Things that are found, genes that are found on the X chromosome. So an example of this, you can look at this, colorblindness. That's a sex-linked disorder. So if you can look at this, and you can see, for example, right there, makes it a 3, or you can fill in this space right here and see that that would form an 8. Okay? If you can see that, and you see this as an 8, rather than seeing it as a 3, might be a colorblindness issue. 5 over here, everybody should be good with that. But you could also see maybe right here, and if that's all you saw, again, might be a colorblind issue where that forms a 2. You might even see it as an 8 if I'm not colorblind, but I think that might actually form an 8. Anyways, red and green are the most common forms of colorblindness. So this is something found, like I said, on the X chromosome. Similar to what we did before, except now the way we treat this is more like X with, for example, a big N or X with a little n. We don't have anything on the Y chromosome. That gene isn't on there, so we don't want to put one of those letters on the Y chromosome saying which type of disorder, what the disorder is. Okay, so for example, let's say we're talking about a scenario where we have color vision, full color vision versus color blindness. Build a Punnett square to show what the children would look like between a homozygous woman who has 2 alleles for full color vision and they have children with a man who is colorblind. So if we look at that, this would be the woman. It said she had full color vision, so she doesn't have the gene, it said in the question, so we wouldn't want to show X big N, X little n. She's X big N, X big N. And then she had children with a man who was colorblind. So that would mean he's got the lowercase n, the recessive allele. And then we do it exactly the same as we did before, except now we've got the sex chromosomes in here. So X big N, that egg, fertilized by that sperm, we get a girl who is not colorblind. She has big N and little n, and the same rules as before. If she has the dominant allele, she won't be having the recessive trait. If this egg is fertilized by that sperm, same thing. Female who doesn't have color blindness. If this egg is fertilized by that sperm, notice there isn't an N on the Y chromosome here because it's not found there. You get a boy who has normal vision. And if this egg is fertilized by that sperm, same thing. We get another boy, XY, who has normal vision. So when we do sex-linked inheritance, it's really important that we kind of change up the focus, the way we do these Punnett squares, and we make sure that we only put that allele, either a capital, a dominant allele, or a recessive allele, on the X chromosomes, and we don't put it on the Y. How are you going to know if it's sex-linked or not? In Science 30, it's going to be a little bit different than Bio 30. In Science 30, we'll tell you. We'll say this trait is a sex-linked disorder, or this trait is caused by a gene that's found on the X chromosome. So it'll be very obvious when you have to do it this way versus back here when you had to do it this way with just the letters. So that's it for genetics. Try some of these practice questions in your textbook. There's a lot of different worksheets and examples, including a bunch of sheets that I've given in class.